I know I've talked about it, but if you don't know, I'm starting a master's program in genetic counseling. If you don't know what that is, I can talk about it another time, but basically my full-time job is going to be teaching people genetics. So I thought this would be a fun little thing to do to get me ready for that. I'm also like relearning a ton of basic genetics in the classes I'm taking, so yeah. But it's just going to be a nice long basic genetics video, and I realized I never actually taught you guys about DNA itself. Like, I've taught about inheritance and genetic diseases and stuff, but not DNA. So we're gonna start there. We're gonna talk about DNA to get us started, and then there will be a lot more information about genetic diseases and inheritance patterns and things like that. So let's get started and let's talk about DNA. I do have a lot of visual aids. I feel like the videos I did where the camera was looking down at me writing didn't do that well, so maybe you guys like this style of videos better, but let me know. So we are going to start over here in the upper corner and talk about DNA. So you may have heard at some point that the structure of DNA is a double helix, and that basically just means that you have two strands and they are wound together in this helical shape. So you can kind of follow one strand going around and wrapping around the other strand. And this is all throughout your body. You have DNA all throughout your body, and it is made up of 4 different nucleotides. The nucleotides are basically just like the letters, the code that make up you. So I like to think of it like a recipe. The letters are like the ingredients that you're gonna need for your recipe, or like the written recipe itself, you know, the letters that make up the words. So we have adenine, denoted by A, thymine, T, guanine, G, and cytosine, C. So your entire DNA makeup is made of those 4 nucleotides. And adenine always pairs with thymine. So that means, like, if you have an adenine on one strand of DNA, then the complementary strand will have a thymine at that location. So you can see that I've color coded, and orange and green are always paired together, and then blue and purple, pink are always paired together, which would be guanine and cytosine. And not super important for like this discussion or what we're going to talk about today, but adenine and thymine form 2 bonds between them, and guanine and cytosine form 3. Again, not super important. It's just based on the like chemical makeup of those nucleotides that they form 2 or 3 bonds. It kind of comes into play with some other concepts, but we're not really going to talk about that today. So yeah, you have a double helix, and in your body, in humans, we have about 3 billion nucleotides. So throughout all of your DNA, in all of your cells, you have 3 billion nucleotides. And that is like, if you're counting the full, like every nucleotide, it would be 6. So like 3 billion is like just one strand of DNA, and then if you're counting the complementary strand, it would be like 6 billion. I don't know. No need to worry about that. And in those 3 billion nucleotides, we have like 20-something thousand genes. We don't actually know the exact number. I've heard 20,000, 23,000, 27,000. I don't know. But those genes each code for a protein. The genes code for all of the things that are happening in your body, and then there's a lot of other DNA, a lot of other nucleotides that don't code for genes, but they are doing other things. So there's a lot of DNA in your body that we don't actually know what it does specifically. genes that code for like liver enzymes. Those obviously aren't going to be expressed and produced in your brain or in your stomach. They're going to be expressed in the liver, and so you need other DNA sequences that make that happen and regulate that. So that's part of what all those nucleotides do that aren't necessarily part of that 20,000 genes. I don't think I needed to go through that fully, but I talk a lot sometimes. Okay, so now let's talk about how DNA becomes the proteins that actually do stuff, because DNA isn't doing anything in your body, it's proteins that are making your body function and grow and things like that. So, the first step in that is a process called transcription, and this is basically taking that recipe and doing something with it, so making the genes. So what happens is the two double helix strands are actually going to separate, and then based on one of those strands, we are going to create an mRNA strand, and that mRNA is going to be complementary to the strand that we are using. And the main difference, well, a couple main differences, mRNA is single-stranded, so it's no longer a double helix, but is just going to be one single strand. The other difference is that RNA does not have thymine, so you don't see any green in the mRNA because there is no thymine. Thymine is going to be replaced by uracil, uracil. So now instead of A pairing with T, A is going to pair with U in our mRNA. So I did actually do this correctly, I think. This would be the complementary strand of this strand on the right. So like we have a T, so in the mRNA we're gonna have an A because A pairs with T, and then like we have a purple C, so we're gonna get a G next, and then an A, we're not gonna get a T, but we are going to get a U. So I wrote out what that mRNA strand would be. And so then once you have this single-stranded RNA, stands for ribonucleic acid as opposed to deoxyribonucleic acid, DNA. Again, not that important, but yeah. So now we're going to do a translation. Translation is the process of actually making the protein from the mRNA. So I guess you can kind of think of it like, you know, you have your recipe. Your protein is what you're making. Your protein is your cake. So this gives you all the instructions to make that. So what happens in translation is that each of these nucleotides are used to tell the machinery in your body what amino acids to add to the protein. So a protein is a chain of amino acids. They're like the building blocks of proteins. And how that is done is that each 3 nucleotides are called a codon, and 3 nucleotides code for 1 amino acid. And so I wrote over here just some random combinations of nucleotides that create different amino acids. So AUG, whenever you have an AUG in the genome, that's an important one. It codes for methionine, and that is what's known as the start codon. So every protein that is made, it starts at an AUG. Methionine is that start codon. So sometimes in some proteins, like that's eventually removed when the protein is processed. You don't need to think about that, but yeah, they all start with a methionine. And then just some random ones like is cytosine. This one, I don't know why I did it like that, but like the U is a bunch of different colors because CCU codes for proline, but CCC and CCG and CCA also all code for proline. So some codons or some amino acids are coded for by multiple different codons, multiple different sets of 3 nucleotides. So that will kind of come in later, like why that's important, but basically there are 64 different ways that you can arrange the 4 nucleotides in sets of 3. 64 ways, but there are only 20 amino acids. So if you do the math, like a lot of amino acids need to be coded for by multiple codons. UAU is tyrosine, and then there are 2 stop codons. They are UAA and UAG, and so similarly to the start codon, the stop codon, whenever you have this, it signals to stop making the protein, that protein is complete, that it's finished, and so the translation stops at the stop codon. Yeah, I think that's all I needed to say about that. So here I've just drawn an amino acid chain. It's kind of random. We'll go into more, like, a little bit more about that next, but amino acid chain, and then after that is formed, you form your mature protein, which is basically just like the folding up of the amino acid chains, makes a protein, and it's kind of dependent on the properties of the amino acids, like how the charge on them or like things like that determines how it is folded. And that could be something like an enzyme that digests your food, or it could be the proteins that make your eye color, like anything. We have a ton of proteins in our body, and they're all coded for by our DNA. Okay, so that was my first page. My second page is going to be about mutations, which are a big thing in, genetics. Like, that's my whole career. In talking about genetic counseling, we're always talking about mutations. So these are just a few different types of mutations that I've drawn out here. So at the top, we have our normal DNA sequence. This is a totally random DNA sequence that I came up with this morning and wrote on a piece of paper. So it can be anything you want. But obviously it's going to start with our start codon, AUG, and we're going to get a methionine out of that. And then we have a proline, a tyrosine, an alanine. These are just like the 3 letters that signify those amino acids. And then a stop codon, UAG. And so that doesn't code for any amino acid, it just tells the body to stop. So we have a chain of 4 amino acids, and that is our normal protein. So now we're going to talk about mutations. So here, everything is the same. All of the nucleotides are the same, except for this guy right there, where we have a U in the normal sequence, and it is changed to a G in the mutated sequence. Well, what do we get out of that? I kind of talked about it earlier, that some codons code for the same amino acid, and so it just so happens that CCU and CCG both code for proline. And so even though we have a mutation and the nucleotide is different, we get the exact same amino acid and the exact same protein. And so that is called a silent mutation, because we don't actually see it. We don't care. It's not causing a change. So next, here, now the change is not that U changed to that G, but it is this C here that is now changed to a U. And so instead of coding for proline, now when you have a deletion or an insertion of a nucleotide. So rather than here where we have like one that's being changed in a frame shift, you would have one that is deleted or added. And so like, let's look at, if we're up here and like we delete this C right here, we're still gonna have AUG, but then the next three nucleotides are gonna be CUU, which is gonna give us the wrong amino acid, and then our next one is gonna be ACG, which is gonna give us another wrong amino acid, and then CCU, which is another wrong amino acid. So if you have that frame shift, you're gonna get a very messed up protein as well, because all of the amino acids are going to be different. So that is a little about mutations. Now, sorry if I'm talking really fast. I feel like I talk really freaking fast. Okay, now we are going to talk about how that all happens in your genome and how like that is passed from parent to child and things like that. So basically we're talking about chromosomes as opposed to just the DNA. So all of that DNA that we talked about, all of those 3 billion nucleotides in your body are organized into 23 unique chromosomes, and you have 2 sets of each of those. So you have 46 chromosomes total, 2 of each of them, so 2 chromosome number 1s, 2 chromosome number 2s, and all of that. And it's basically all of this coiled DNA. And so they are organized of like all of your 20-something thousand genes, so maybe 1,000 of them are on this chromosome, and then 2,000 of them are on this chromosome or whatever. Like, it's all of the genes organized in your body, in your DNA. So yeah, you have 22 pairs of what are called autosomes, which are like chromosomes that both males and females have. You get one of them from your mom, one of them from your dad. And then there are sex chromosomes. So that is the only difference between males and females as far as your DNA goes, is the sex chromosomes. And so I drew these. This is an X chromosome, and then a Y chromosome is a lot smaller. It has a lot less genes. And females have 2 X chromosomes, so they get an X from their mother and from their father, and then males would have an X and a Y. So because females have 2 X chromosomes, they always pass on an X chromosome to all of their children. Like, you can only pass on what you have. You can't pass on something you don't have. So they're always going to pass on an X, and that's why females and males both get an X. But males have an X and a Y, and so they are getting their Y from the father. So like, a female always passes down an X, and then the dad has a 50% chance of passing down his X, which would make a girl, or his Y, which would make a boy. And I feel like I always say it in these videos, but like, I am talking purely about genetic sex. I fully support anyone that does not identify with the gender that they were born as, but when you're talking about genetics, a female has 2 X's, a male has an X and a Y, even if that's not how they identify. That's how your assigned sex at birth is determined. Yeah. So, we've got a female who has 46 chromosomes total, an X and an X. So that's how you would write the notation for a female, and then a male would be 46XY. And we're going to talk about this meiosis. So basically, everybody has 46 chromosomes, and when you are making your sperm or egg in the process of having a baby, your cells are separating, and so each sperm or each egg is going to get 23 chromosomes, one of each of those pairs. So one chromosome number one chromosome number 2, one sex chromosome, X or Y. And so that's why I wrote here 23X. So 23 chromosomes. The sex chromosome is an X. And then the dad is also making a 23X. So this is an egg and a sperm. And we get a baby who now has 46XX. An X from each parent, 23 chromosomes from each parent. So now they're 46XX, and that is a little girl. So that's the normal way that happens. But we're going to talk about an issue that can happen in meiosis, and this is how you get chromosomal abnormalities. So we talked before about point mutations in the DNA sequence, whereas chromosomal abnormalities are issues with like how many chromosomes you have. So they're generally like a lot larger issues. But so, like I talked about over here, these would be like the cells in the mom and dad's body, germ cells specifically, if you want to get specific. But so each of them has 2 of each chromosome. Like, let's say that the orange one, for purposes down here, we're gonna say the orange one is chromosome 21, and then the green one is chromosome 3. I don't know, it can be whatever you want. But so then to make eggs and sperm, this has to separate, and ideally each egg and each sperm would get one of each of those chromosomes. But what happened here, you can see, is something called non-disjunction. And so this is where both of the mother's orange chromosomes went into one egg cell. And so by default, the other one didn't get any orange ones. They got the normal number of green. They each got 1 green, but this one has an extra orange one, and this one has none. And so the male, everything happened normally. Each one has 1. But now when we go to form a zygote or a baby, now if this egg merges with this sperm, we are going to get only 1 orange chromosome, because this one has none, this one has 1, so just 1, and then our normal 2 green ones. And this is not going to form a living baby. There are no monosomies, is what it's called when you only get 1 of a particular chromosome. No monosomies are compatible with life, so this would be, it either would never get implanted or it would be a very early miscarriage. But down here, if this egg merges with this sperm, we're gonna get 3 of the orange one and 2 of the green one. And so most of the time this is also not compatible with life, but an exception is Down syndrome. So that's why I said the orange one would be chromosome 21, because if you have 3 of your chromosome 21, you're going to have Down syndrome. And we know that people with Down syndrome can live long, healthy lives, so relatively. And so, yeah, that is how Down syndrome can happen, is a non-disjunction of the 21st chromosome. And yeah, it can happen. Chromosome 13 and chromosome So Down syndrome, trisomy 21, is the main trisomy that is compatible with life. And here we've got a girl with Down syndrome. Because of this, she ends up with 47XX. So she has the normal 2 of every other chromosome, but she's got 3 of the 21st, which gives her 47 total. Yeah, so that's chromosomal abnormalities. It's how monosomies, trisomies happen. Yeah, kind of a fun fact is like a trisomy can happen in any chromosome. You can have trisomy 1, trisomy 2. It's just as likely for that to happen. It's just that those babies are not compatible with life. It sounds horrible, but like that's the terminology that you use. It's compatible with life. So they're just like miscarriages, so you never knew that it happened. But it is just as common. It's just that the 21st chromosome is actually your smallest chromosome, and so if you have 3 of it, it has the least effect. You can still survive, whereas if you got an extra of your first chromosome, which is the first or the largest one, that would be like such a large impact that the baby would not survive. Okay, so now we are going to talk about inheritance, and like dominance and recessive traits and all that jazz, which is the part of genetics that I like. Yeah, so we are going to start with not talking about humans. We are going to be talking about pea plants. Pea plants. And the reason we're doing this is that that's the first organism that genetics was really studied in, and that was by Gregor Mendel. He is known as the father of modern genetics, and he studied pea plants and basically discovered genetics. And a fun fact about that is that Gregor Mendel was a monk. He was a monk, and he originally wanted to study mice. He wanted to like study genetics in mice, but he was told that because he was a monk, that would be inappropriate because mice have sex. And so he went with pea plants, and it's kind of funny because if he had studied mice, he probably would not have become the father of modern genetics, because mice are a lot more complicated. Like, if you think about humans, we don't have many traits where it's like, okay, there's this and this. Like, you're not tall or short or like blue eyes, brown eyes. Like, there's such a spectrum with everything. Plus, there's a lot longer generation time and like less offspring, so it's really hard to study that. Whereas pea plants, they can be green or they can be yellow, they can be wrinkly or they can be smooth, and they produce so many offspring, so it was like really easy for him to study genetics. So yeah, because he was a monk, he studied pea plants and he became the father of modern genetics. Anywho, so we are going to start with talking about just one trait. So as I said before, the pea plants could either be green or yellow. And so what he did, so this YY, that is known as the genotype. So it's the set of genes, set of alleles that an organism has. And so like when looking at our chromosomes, let me get my paper back. So like let's say on this chromosome you have like a Y, a big Y on this chromosome and a little y on the other counterpart, like on your 2 first chromosomes. If that's what you have, then your genotype would be big Y, little y. You're getting a big Y from one parent and a little y from another parent. So yeah, that's genotype, is the alleles, the genes that you get from your 2 parents. And yellow pea plants are dominant, and so that's denoted by recessive is green, and so you always use the lowercase version of the dominant letter. So we have lowercase y's here. So that's the two genotypes, and then phenotype is what is actually observable. So like the phenotype would be green and the phenotype would be yellow. The genotype is big Y, and genotype is little y, little y. Yeah. Okay, so then we're going to draw a Punnett square. A Punnett square is basically how you determine the possible offspring that two parents could have. So what you do is you separate the genotype. So here we've got our yellow pea plant on the top. So we have a big Y and a big Y, and that is like that because those are the two alleles, two genes that parent can pass on. They can either pass on this big Y or this big Y. And then same with the green one, they can pass on this little y or this little y. And so because both of these are what we call homozygous, homozygous means that they have two of the same allele. So two big Y's, two little y's. And when you have that, all of your offspring are going to be heterozygous. So heterozygous meaning one big Y, one little y, because all of this parent can pass on as a big Y and all this parent can pass on as a little y. So they're all going to get the same thing. And because yellow is dominant, all of these are going to be yellow, all yellow offspring. So a recessive trait, you have to get both recessive alleles. You have to have two recessive alleles in order to show that trait. Whereas for dominant, you can be homozygous for that dominant allele, or you can be heterozygous and you will still show the dominant trait. So basically, all you need is one big Y to be yellow. You can have two, or you can have one, and you'll still be yellow. So that's what we have here. But then next, what you do next is you cross two pea plants from that generation. So two of these child pea plants you then cross. So they're both yellow and they're both heterozygous. And if we do our Punnett square now, we see that if each of them pass on a big Y, we will get a homozygous dominant. If one passes on a big Y and one passes on a little y, we will get heterozygous. So they will still be yellow, just like their parents. But both of them could pass on the little y, which we would now get a green pea plant, just like the grandparent. And so this is basically how he figured out genetics, by showing that two yellow pea plants could produce a green one, which is kind of like counterintuitive. It's kind of interesting, like two parents with brown eyes having a blue-eyed child. It's totally possible because of this. So basically showing, I think it was previously thought that like you were like a mixture, a homogeneous mixture of your parents, which would kind of mean that like you couldn't, you know, if you had two yellow parents, like you were gonna be yellow, or if you had a blue and a yellow parent, you're gonna be green. Like you're gonna be one trait that's in the middle of them. But this proved that wrong in that we can see this phenotype again, even after like eliminating it in this generation, it comes back. And that's because these alleles are independent of each other. They independently segregate. And these are just the ratios. So as for genotype ratio, we have one homozygous dominant, two heterozygous, and one homozygous recessive. So 1 to 2 to 1. But then phenotypic ratio ratio is going to be 3-1, so 3 yellows, 1 green. Yeah. The next thing I'm going to talk about is when we actually have 2 different traits that we are looking at. So, in addition to yellow and green, pea plants can be smooth or wrinkled, and smooth is dominant to wrinkled, or round. I guess smooth, round, same thing. I'm using round because, yeah, R. So if we have a yellow round and a green wrinkled, and we cross those, again, we get heterozygous for both, just like over here. So yellow and round, but heterozygous. And then here we do a big old Punnett square, where we're talking about both traits. So these now, it's like this parent, you have to think of this like 2 parents, like here, but they have the same genotype. But like, so this parent could pass on a big Y and a big R, a big Y and a little R, little y and a big R, or little y and little r. So it looks more complicated than it is, but it's basically just like doing this for 2 different letters, and this is what you would get. So you would have 9 individuals. These 9 that are yellow would be yellow and round. They have different genotypes, but as long as they have 1 big R and 1 big Y, they're yellow and round. And then you get 1 that is green and wrinkled because they got all heterozygous. But then the red ones, or orange, I don't know, are yellow and wrinkled. Yellow and wrinkled. And then the blue are green and round. So, yeah, you get a lot more combinations in a 9-3 to 3-1 ratio. Yeah. I just want to get to the humans part, so like I'm kind of rushing through this, I guess, but yeah, that's what you do. And then I just drew this over here. I know I kind of mentioned this before, but it's just something called incomplete dominance. So normally for most traits you have like green and yellow, and heterozygous is going to be yellow. It's recessive and it's dominant. Whereas here with incomplete dominance, a homozygous dominant, let's say, is blue, and a homozygous recessive is yellow. But you would think a heterozygous would be blue, because they would get 1 big B, but the heterozygous is green, which is a mix of those 2, a mix of blue and yellow. So that's called incomplete dominance, where you do actually get that mixture of the 2 traits. So yeah, you can see that if you have 2 heterozygotes, you're gonna get a blue, 2 green, and a yellow, just because that's what the genotypes are. And so you can kind of think of that like height or something, where maybe like if one of your parents is really tall and one of them's really short, maybe you're gonna be in the middle. Like you're not gonna be tall or short, but maybe in the middle. So it's way more complicated than that, but like that's one way that you can think of it. And we are kind of going to be seeing incomplete dominance in our next example that I'm going to talk about. So now we are moving on to humans, my favorite topic. I like talking about actual human genetics, and we are going to talk about an example and kind of pull everything together that we have been talking about. So we are going to learn about sickle cell anemia, and this will include Punnett squares, point mutations, all sorts of things. Okay, so it does get a little confusing when you're talking about diseases, because if you're talking about a recessive disease, then dominant is normal. So I know like with the other ones, it's like, oh, if you're dominant, you're yellow, but like dominant is the absence of the disease, and then recessive is the disease. So that's a little like, you have to think about that. But so the normal allele. So an allele, I guess I didn't explain this that well, but like an allele is the version of a gene that you get. So you have two copies of each gene, one on each, like a gene on your first chromosome, you have two copies of it, one on each of your first chromosome that you get from your mom and your dad. Yeah, so gene copies. An allele is which gene copy you get. And so the normal allele, the one that you're supposed to have that is not sickle cell anemia, it's a much longer sequence than this, but the part that is actually changed in sickle cell anemia is this particular codon, which is supposed to be CTC. CTC, which when it's transcribed into mRNA, you get GAG. And so when you make a protein, you get a glutamine, I think, glutamine. And that, when you have this normal protein, you get normal red blood cells that look like this. However, if you have the sickle cell allele, then the rest of your protein is normal, but at this CTC site, you have CAC. That's the point mutation. This is known as a missense mutation, which we saw before. And so when it is transcribed, you get GUG, and that now codes for a valine. So again, missense mutation. We have a different amino acid than what we want, and that leads to a misfolded protein, and it leads to red blood cells that look like this, which is known as a sickle shape. And that's where sickle cell anemia comes from. And basically what causes your symptoms is that those sickle-shaped red blood cells can get like caught together and get caught in your blood vessels, and then that leads to pain and all sorts of symptoms. So that is sickle cell anemia. And so over here, if we draw a Punnett square, let's say we have two individuals, mom and dad, and they are both heterozygous for the sickle cell trait. And so if sickle cell is recessive, it means that we would need both sickle cell alleles in order to actually have sickle cell anemia. So these two individuals do not have it. They have the dominant trait, which is normal, but they are carriers of that sickle cell allele. They both inherited a sickle cell allele. And so when they have a child, 25% of their children will be completely normal, no sickle cell allele. 50% will be carriers just like them, but 25% will get the recessive allele from each parent and will have sickle cell anemia. So that is how a recessive disease is passed on. And just something interesting about sickle cell anemia. The reason that it is prevalent, and it is more prevalent in African Americans or African individuals, is because sickle cell actually protects you from getting malaria. So while sickle cell is not good and causes a lot of issues, those sickle-shaped red blood cells can't carry malaria, and so these individuals are resistant to malaria. And so that's why sickle cell actually evolved and spread in Africa, where malaria is really common because it protects you from it. So yeah, whereas like in places where there is no malaria, like it's just a disadvantage to have sickle cell, whereas in Africa it's actually kind of an advantage. And reason I said that that's kind of incomplete dominance is that if you are heterozygous, you won't have sickle cell disease, but that having that one recessive allele does actually produce some sickle-shaped red blood cells, which makes you more resistant to malaria. So heterozygotes they don't have sickle cell, and they also can't get malaria, and so that's super advantageous. But then obviously if two carriers get together and have a child, they could have a child that has sickle cell anemia, but it just shows how it's an adaptation and how how sickle cell happens and why it stays really prevalent. Okay, next we are going to talk about pedigrees. So this is kind of the end of what I'm going to talk about today. Pedigrees are a huge thing for genetic counseling. It's basically a family tree, a family history, and we are going to be talking about pedigrees. Okay, so this is what a pedigree looks like. We have two different pedigrees here, one on top and one on the bottom. And like I said, it is a family tree, so circles are females, circles are the moms, squares are males, the dad, and then we have, so if there is a horizontal line connecting two people, they are married, so this is husband and wife, and then the lines coming off of them are their children. So these two parents had 4 children, and then their son married this woman and their daughter married this man. So that's how you would read a pedigree. And the filled in shapes are affected by whatever disease you are looking at. So you draw these when you're thinking about a disease. The filled in ones have the disease and the blank ones do not. So a big thing that we do in class is trying to determine the inheritance pattern of a trait based on the pedigree. And so looking at this one, what we know is that we have 2 unaffected parents, parents that do not have whatever disease we are looking at, but they have children that are affected. So immediately, that signals that we are looking at a recessive disease, because like I showed, you can have 2 parents that are both carriers, so they don't have the disease, but they have children that do get the disease. So that's what we are looking at up here, is a recessive pedigree. And then, so with recessive, it is possible for them to have children that are not carriers at all, or to have kids that are carriers, or to have kids that are affected. So we can see that these 2 kids are affected, and I drew the Punnett square for that up here, so homozygous dominant, and then heterozygous and homozygous recessive. They had 2 affected children, 2 unaffected children, and this unaffected child married an unaffected man, and they had children, but one of their children got the disease. So we know, looking at that she must be a carrier. She must have gotten one sickle cell gene from one of her parents, and her husband must also be a carrier. If someone's a carrier and their spouse is not a carrier, they can't have a child with the disease. They can have children that are carriers, but not children with the disease. So because they had one with the disease, we know these must also both be carriers. Over here, we have a child who's affected who marries someone that is also affected. And so all of their children are going to be affected, because all they can ever pass on is the sickle cell gene. That's all they have. So all of their children are always going to be affected. I should have drawn another example where person married someone that was not a carrier, in which case all of your kids would be carriers. None of them would have it. All of them would be carriers. But yeah, so that's how a recessive trait shows in a pedigree. I don't think I'm really going to go into this because I didn't talk about dominant traits at all, really, but this is a pedigree of a dominant trait. Yeah, which basically means like you only need one allele to have the disease. And I'll just say quickly that like the reason we know this is dominant is that 2 affected parents had unaffected children. So like this child of theirs is unaffected, which means like if it was recessive, then 2 affected parents could only ever have affected children. But like if they are both heterozygous, they can have a child that's homozygous recessive, which means they wouldn't have that trait. I feel like I'm not explaining things that well. I don't really I draw things out, but I don't plan what I'm gonna say, so sometimes it just comes out wrong. But I hope you guys are enjoying this. But yeah, the last thing I'm gonna talk about is X-linked or sex-linked traits. So what we were talking about was all autosomal traits, meaning that the genes are located on autosomes, chromosomes 1 through 22. So males and females have equal likelihood of being affected. But now we are going to talk about sex-linked traits. So as we talked about, females have 2 X chromosomes, 2 X's paired. A male has an X and a Y, and the Y doesn't have a lot of genes on it. It basically just has the genes that make you a man, but not like other genes. So essentially, for this chromosome, females can be heterozygous, like they can get one allele on one X chromosome and one on the other, but males are always homozygous. They only get one X chromosome, so they only get one allele, and that's just the trait that they have, is the one allele that they get. So yeah, that creates gender differences when we're looking at traits. So one trait that is X-linked or sex-linked is color blindness, meaning that the color blindness gene is on the X chromosome. And so for this female, we are saying that pink is normal, not colorblind, and green is the color blindness gene. So the female has one of each. She's heterozygous. And because color blindness is recessive, this female is not going to be colorblind because she has a dominant allele which takes over, and she will be completely normal. Whereas for the male, he also has one color blindness gene just like the female. However, because he only has one X chromosome, he doesn't have any other one to mask it. So he doesn't have a dominant allele, so he's gonna be colorblind. They both have one recessive allele, but only the male is colorblind. And this is why colorblindness is a lot more common in males, is because it's sex-linked. So yeah, that's what we get. And so if we look at I almost said pedigree, a Punnett square down here, so this is how we draw the alleles, is like the mothers would be X with a big B, the normal, and then X with a little b, which is colorblind because she's a carrier, and then the male has X with a little b and a Y, so that's what they can pass on to their children. And so here we would get, these 2 would be females because they have 2 X's, and these 2 would be males. And this female is a carrier. She's a carrier for the trait, so she doesn't have color blindness, but she's a carrier. And then this female would have color blindness. Because if we are looking here, so here we have a carrier female, big B, little b. She's not colorblind, but she's a carrier. And then a normal male, he is not colorblind. So neither parent is colorblind. Here we are gonna get a completely normal female, not a carrier. A carrier female. So none of the girls are going to be affected with color blindness because they're all gonna get the one X that the father has. And if the father's not colorblind, he cannot pass on the color blindness gene to his daughter. And so she can't be colorblind. She could be a carrier, but she's not going to be colorblind. Whereas the boys, we are gonna get a normal boy from the mom's normal gene, but we are also gonna get a colorblind boy because the mom can pass on that recessive allele, and the boy only gets one, and so he's gonna be colorblind. So yeah, basically the tagline, or I don't know, whatever, the takeaway for X-linked traits is that in order to have an affected female, she must have an affected father. That's the big rule. If a father does not have an X-linked trait, the daughter can't get it. The son could get it, but the daughter cannot. And here I just showed the same for an unaffected female and an affected male, and none of the kids are colorblind, even though the father is affected, because the female always gives an X to everybody, and if she is not a carrier, then everybody's going to be at least heterozygous or completely normal. So yeah. And this is just a pedigree showing an X-linked trait. So basically what we see is, like, anytime there is an unaffected father, so like this father's not colorblind, this one's not colorblind, they will never have affected girls. So no affected girls, no affected girls, only a boy that's affected, meaning she's a carrier. That's what this means, the fact that he's affected. And then here, when we see that they have kids that are colorblind, it means she must be a carrier. Because like we saw down here, if she's not, none of the kids would be colorblind. And then yeah, over here still an affected female. All of her boys will be colorblind. Because she's giving them their only X, and she's colorblind, so they will be colorblind. But the dad is not, so the females will all be heterozygous carriers. Yeah. I hope that that was an okay explanation of genetics. Yeah, we learned a lot today, so try to digest. Let me know if you want to see any further genetics videos. I do really like making them, but it did take a lot of work. So yeah, definitely took a lot of work to put all of those sheets together, and I've been talking for so long, so my throat hurts, but yeah, I hope you enjoyed this. Sorry for my talking 12 miles a minute.